floating in the fish. It is one molecule. But if we want to study how the bonds occur in the fish, what we can do is boil this under non-reducing conditions, and weak bonds will break away if they don't have a disulfide bond. So let's picture I boil my antibodies. What's going to happen here? It's still going to remain fully bound. However, if I boil this antibody, what do you think is going to happen? This breaks away, we get a monomer, we also get a trimer. In this case, two are going to break away, so we get two monomers, and we get a dimer. But, as I said, it's important that you realize that in the fish, it's not like these different structures are floating around in the system. You always have this one bound molecule. Now, if we run these different structures after I boil on a gel, what you can see is you get four banding patterns. You get the tetramer, trimer, dimer, and monomer in fish. In contrast, the mammalian IgM is much more simple. It's always fully bound at all times. So you just get one band, even if you boil it. Now, it's predicted that this differential cross-linking could be an alternative to the isotypes that the mammals had. So perhaps rather than relying on IgG, they rely on a fully cross-linked molecule. Maybe instead of IgA, they rely on a molecule that has less cross-linking. So that in essence, it does have different isotypes. Now, what's also come in the literature, actually in 2010, is that fish actually do experience affinity maturation. That is the increase in affinity of the antibody. And also, you get an increase in the degree of cross-linking throughout the immune response. That is, you go from low cross-linked antibody that's flexible to highly cross-linked antibody that's rigid. And to study this, the researchers used a very artificial antigen known as TMP-KLH, but it has the advantage that it gives a very defined response because there's only one epitope to which a B cell can bind. Pathogens, on the other hand, have hundreds if not thousands of epitopes that B cells can bind to. Now, following vaccination with fish, they studied how cross-linking and affinity to TMP-KLH changed throughout the immune response. And what you can see here, it's kind of hard to see, but these white boxes are tighter. And it increased nearly 1,000-fold throughout the immune response at its peak. Now, affinity, on the other hand, increased about 2 logs, or 100-fold. Now, this isn't the levels that we see in mammals, but you need to remember that IgM is a tetrameric molecule. So small changes in affinity can lead to large changes in the sum of affinity, known as avidity. Also, what they found is, as I said, the degree of cross-linking became greater throughout the immune response. So just focus on the tetramer here in these gray boxes. That is the fully cross-linked form. What you can see is it keeps increasing throughout the immune response. So that towards the end of the immune response, it made up about 50% of all antibody. Now, what you're probably wondering though is, are these studies with a simple artificial antigen actually even relevant to aquaculture? Can we transfer this knowledge to pathogens? And even if we can, what is the biological significance? And this is where my research came into play, bridging the gap between what was known in artificial antigens to actual pathogens, which, as opposed to being very simple, defined, are very complex. So think of TMP-KLH as being one key, one lock. In my case, we're studying multiple keys, multiple locks. So a change in a key is going to be could be very difficult to determine. So my model organism was Streptococcus iniae. Now, Streptococcus iniae is a gram-positive bacteria, and all I'm going to say about that is we use a gram-positive as opposed to gram-negative because it gives a robust immune response because you have T-cell activation involved. Now, it's been described as one of the most formidable pathogens of the last decade. And just from a local perspective, we actually have, in Massachusetts, the largest aquaculture facility in the world. And in the late 90s, Streptococcus iniae shut down that facility that was growing striped bass because it devastated it. Now, this summer, well, they had reopened and then they were growing barramundi instead. This summer, our lab actually got called in because they had another outbreak of Streptococcus iniae. Now, in 1997, SNEA resulted in $100 million of losses globally. And death from SNEA in fish is usually a result from meningoencephalitis, that is, swelling of the brain. And the reason I'm using SNEA is because the knowledge is transferable to other pathogens like Legionella and Vibrio. Now here's the aquaculture facility I was talking about in Massachusetts. Each of these is about 3 stories high, each of these tanks. And as I said, SNEA shut this down. Now here's one of my fish that was infected with SNEA. And as you can see, the eyes are bulging out of the head. They literally pop out of the head following vaccination. And this is because you get immune cells like multiplying behind the eye, and then you get recruitment of other cells, you get inflammation. Now, my model organism is the rainbow trout. And the reason that we use rainbow trout is it's highly desirable. I mean, your mouth is probably watering just looking at it. And it's also highly commercial. That is, it's desirable in aquaculture. And I can apply my knowledge from the species to other fish, freshwater and marine species. So my objectives were to determine, following vaccination with killed SNEA, how the quantity, specificity, and quality being degree of cross-linking and affinity of IgM, specific IgM, changed throughout the immune response. And my predictions were based on what's in the literature. That is, I expected to see the titer to increase, the repertoire specificity of the proteins to increase, degree of cross-linking, and affinity to increase. I also thought affinity and cross-linking would be correlated, and that was based on literature as well. So how did I address this? Well, I vaccinated 150 fish with my killed bacteria, and then up to weeks 37, I collected 500 microliters of blood from the fish per time point. I allowed the blood to clot overnight, and then I collected the serum. And the serum is what contains antibody that's both specific and non-specific to my vaccine. And then I later assess this for quantity, specificity, and quality. So first, I had to determine the titer of how the titer changed throughout the immune response. Now, to do that, we use a common technique in immunology known as the ELISA. And it's performed on a 96-well plate. We're going to focus on a single well of that 96-well plate. So I coat it with my bacteria, what I vaccinated the fish with. I introduce the serum, and any antibody that's specific to my antigen is going to bind. Any that's not specific will be washed away. I come along with another antibody that will bind to my bound fish antibody, and attached to it is an enzyme that will react with the solution to produce this green colorimetric change. Now, the greater the titer the fish has, the greater the green travels along the plate. Now, by eye, I can see this fish has a greater titer than this fish. To actually quantify that, we use a plate reader. And then I get my ELISA curves. Now, we determine titer by the 50% point where these curves cross. The only reason we do that is because it's the most sensitive point of a curve. Now, all I want you to know is that the farther along the curve travels to the right, the greater the titer a fish has. That's all you need to know, unless I can quantify that. Now, I also had to determine how the specificity of antibody changed throughout the immune response. Now, to do that, we did a process known as Western blot. So what I do is I take my antigen, again, SNEA, what I vaccinated the fish with, sonicate it, that is, I shatter it. Then I run it under reducing conditions, which breaks away the disulfide bonds, linearizes it. Then I run it on an SDS page gel. And each band here represents a protein that the immune system can recognize. So there are many proteins that the immune system can recognize, and within each of these bands are many epitopes that an antibody can bind to. I then transfer that gel to a membrane. And at this stage, you can't see my protein that's been transferred. All you can see is this marker, and the marker just allows me to determine the molecular weight of the proteins. I then incubate that with my antibody, and I do this on a slot blot apparatus. The slot blot apparatus just allows me to use many different samples of serum on a single membrane. Now, any antibody that's specific to a protein on that membrane will bind. Anything that's not specific will be washed away. Now, after that, I add an antibody that will bind to my specific fish antibody and is conjugated with a fluorescent tag. Because of that, I can detect it with a fluorescent scanner. What's important is that you realize that each of these lanes represents a single serum sample. And within each of these lanes, you see many different banding patterns. And each of these bands represents a protein that the fish antibody has bound to. Now, what you can see is that for this particular time point, the banding pattern is about similar from fish to fish. But there are individual differences. So if you look here, these two proteins are only recognized about by 2 fish. So there are individual differences. All right. I also had to determine how the quality of the antibody changed throughout the immune response. By quality, I'm referring to degree of cross-linking. And I did this for total antibody, which contains both specific and non-specific antibody to my antigen. I also did it for specific antibody, that is just specific to the antigen. And the way I isolate these is by, very similar to the ELISA, incubate it with my bacteria, these will bind to the bacteria, and I wash away unbound antibody. From there, I boil it as we described at the beginning of my presentation, and what's going to happen? You're going to generate these different structures, the tetramer, trimer, dimer, monomer. I then run that on a gel, and each band again represents the different structures, tetramer, trimer, dimer, monomer. And they range in weights from high to low. Now from I, what you can see is that the intensity of this is far greater than this. That is, tetramer makes up a greater proportion than monomer does. But I can't quantify that by eye. And that's where densitometric analysis comes in. I use software to determine the intensity from each of these bands. So I just highlight it with a box with the software, and it gives me a readout. And from there, I can very simply in Excel figure out the proportions that each of these bands makes up. So for the total antibody, tetramer made up about 42%, that fully cross-linked form. And specific made up about 40. And then I can take the ratio of specific to total and determine how that changes throughout the immune response. Now, I also had to determine, lastly, fortunately, how affinity changed throughout the immune response. And this is very complicated, so I'm going to keep it simple. Let's imagine we have a high affinity antibody and a low affinity antibody. If we incubate that with bacteria, it's going to come to equilibrium. Now, this that binds with high affinity antibody is going to tend to be like a lock and key. It's going to tend to be bound at all times. This low affinity antibody is going to have a high on and off rate. So it's going to go on, off, on, off. So its sites mostly are going to be unbound. Now, because of that, I can determine differences between the antibody. So this high affinity antibody is going to take less bacteria to inhibit it than this low affinity antibody, which is going to take a ton of bacteria to get all these sites bound. Now, using an ELISA that I described before, I can then see how this changes throughout the immune response. Now, in this case, it's the opposite of probably the way you're thinking. The farther along the green travels from right to left, the less affinity that particular sample had, because it's an inhibition process. Well, all you really need to know is that from the ELISA, I get these inhibition curves. And thus, what I can say is this curve here, which took less bacteria to inhibit it, has a greater affinity than this green curve here, which took more bacteria to inhibit it. So thus, this red sample that's at 0.2 has 5 times the affinity than this one that crosses at 1. On to results. So when we looked at titer, that is the quantity of antibody, what you can see is that by week 5, we got about a 500-fold increase, 200-fold increase, in antibody levels. Week 0 and 2 were about the same. Week 37, the titer did begin to drop, but it was still 23 times the levels of the starting. I then looked at how specificity changed, and I'm just using one sample. I'm just showing